Dear colleagues, welcome to our Clinical Trials Masterclass. It's the fourth lecture we have today, and it's the first lecture of the second module. We completed the first round of the first module, which focused on heart failure, and are very happy to discuss the topics regarding coronary artery disease with a wonderful guest. And I would love to introduce my co-host today, which is Aisha Kader, interventional cardiologist from Dhaka, and Aisha is a fellow student in the master's program at the University of Oxford, focusing on clinical trials. Dear Aisha, welcome to our masterclass.
Thanks, Jawed. And it is my absolute pleasure to introduce Dr. Roxana Mehran, who quite honestly needs no introduction, but seeing as I'm duty-bound to provide one, she's an internationally renowned interventional cardiologist and cardiovascular clinical trialist. Dr. Mehran is professor of medicine and cardiology and population health science and policy at Mount Sinai. She has built a globally respected academic research center focused on developing randomized clinical trials and has served as PI for numerous global studies, including the Science Short Depth Program Science 1928 and the TWILIGHT trial, which she will discuss shortly. She's developed risk scores for bleeding and AKI and has authored over 1,000 peer-reviewed articles. Dr. Mehran is the recipient of numerous accolades, including the 2018 Nanette Wenger Award from Women's Heart for Excellence in Research and Education, the 2019 Ellis Island Medal of Honor, and the 2019 ESC Silver Medal and Andreas Grunzig Lecture Plaque, which I had the honor of viewing in person. She has been included for 3 consecutive years in Clarivate Analytics as the top 1% of the most cited researchers, as well as being included in the world's most influential scientific minds by Thomson Reuters. Also, in 2019, she founded Women as One, dedicated to advancing opportunities for women in medicine and cardiology, of which I myself am a grateful beneficiary. Dr. Mehran, we're so thrilled to have you with us today, and we're really looking forward to learning from you. Take it away.
Thank you, Dr. Kader, Dr. Hashemi. It's really just wonderful to be with all of you. I must say I'm a little bit nervous about this wonderful opportunity, and I hope that I don't disappoint. I think one of the things we want to do today is in kind of taking you through my journey of the TWILIGHT trial, and I'm about to share with you. Can you guys see my slides or not yet?
Not yet.
Okay, let me share my screen. Sorry. I'm about to share my slides here. How's that?
Perfect. Very good.
Excellent. So without further ado, let's move towards this class on designing trials in antiplatelet therapy. This is not a small task, extremely difficult, and I'm humbled to be asked because there are so many incredible leaders who have done beautiful work in designing some of the most important randomized clinical trials in antiplatelet regimens, but some insights from the TWILIGHT trial. Before I start, it's important for you to note my disclosures. As a head of an academic research organization here at the Icahn School of Medicine, my institution and my center is recipient of multiple awards from device and pharmaceutical companies, and important for you to note my roles here. But in terms of the TWILIGHT trial, let it be known that this was sponsored by Icahn School of Medicine at Mount Sinai. I was the sole applicant as the responsible party for the TWILIGHT trial, and it was through a significant grant from AstraZeneca. So let's begin. And for those of you cardiologists, I apologize, but I want to make sure that everyone who's in this class understands the dilemma that we face in designing clinical trials in acute coronary syndromes or coronary artery disease, where we are looking at different syndromes, different pathobiology, and the treatment options that then bring us to drugs, namely antiplatelet regimens that have great benefits, but also have side effects and important harm of bleeding. And so as a background, the syndromes are here: acute coronary syndrome, one that is about a plaque rupture thrombus formation versus a chronic coronary syndrome that occurs in patients who have risk factors with blockage and the atherosclerotic plaque limiting blood flow, usually those syndromes being that of exercise or stress, etc. Whereas an acute syndrome is mostly associated with injury to the myocardium, heart attacks, etc., and thrombus formation. For the most part, a lot of, for acute coronary syndromes, percutaneous coronary intervention early on, especially for MI, has been extremely beneficial with the use of a stent, which is a metal cage that goes into the artery, opens and gives you blood flow. But in combination with what we call dual antiplatelet therapies, which is aspirin plus another agent, a P2Y12 inhibitor. Let me just tell you, why are they needed? Why do we need these drugs? Because, as I mentioned, both in the syndrome and then after you treat the patient with a metal cage, platelets play a very, very key role in not only that stent failing, because it's a foreign metal cage inside of a small artery, so a nidus for thrombus formation, so also associated with stent-related complications, such as what we call stent thrombosis, but also that same patient is at risk for non-stent related ischemic events, such as recurrent myocardial infarction or a stroke. And so antiplatelet therapies have had a pivotal role as the cornerstone therapy for our patients. But over the years, there's been an evolving story about antiplatelet therapies, depending on what's going on with everything else that's going on in development of our devices, improving and enhancing our techniques and our ability to now use the next generation devices. So where in the early years, in the 90s and the early 2000s, everything was about the stent clotting off. Once our stents got better, our technology improved with thinner struts and next generation of stents, and we began to even have better antiplatelet regimens, more potent regimens. We became more aware of the bleeding side effects because the ischemic events were no longer as prominent, and we started to see the bleeding events. And now we are starting to think about, in this bleeding arena, which was a couple of decades ago, and I'll say that I had a lot to do with this because we started seeing bleeding as a very important association to mortality. And now where we are is in this sort of a equipoise of both of these being very, very important. So if you're designing a clinical trial for antiplatelet regimens, you really are in a dilemma. In a dilemma because the devices have gotten a lot better, but the patients have also become more complex with more comorbidities, and therefore are at a higher risk for ischemic events, may not all be related to the stent that you're placing in the small coronary artery, but the patient is older. They have more chronic kidney disease, more diabetes. They're coming in sicker, more complex. Then that same patient is also at risk for bleeding events. So that calculus of figuring out where is the sweet spot of finding the best possible place to give a patient an ischemic protection of a drug without having a harm is something I would say is the golden goose, or the place we all want to get to. And as a result, you will see that the guidelines have all kinds of different permutations of what to do depending on the patient's bleeding risk, high bleeding risk, low bleeding risk, what duration of antiplatelet regimens. And at the end of the day, the clinician is still in implementing the science that goes behind these guidelines based on clinical trials, have to apply it on patients. So when you're designing a study, it depends on where you are at the time and what are the guidelines are saying, and what are some of the possibilities of where we are at the time as you're designing that study, of the unanswered questions that you want to help clinicians answer in the ways to improve the clinical events and the outcomes of the patients overall. And that brings us to what are some of the things that we could do? Well, what can you test in antiplatelet therapies? Lots of things, comparing two different drugs, seeing if an additional drug would work better than an old drug, or can we add another drug that could also be on top beneficial? This is something we love to do, keep adding drugs. Can we bring down a potent agent to a less potent agent, de-escalate because we're worried about bleeding? Can we test different durations? Maybe we don't have to give 12 months. Maybe we go to a 3-month or a 1-month duration in different kinds of scenarios. But what about something really out of the box? What about taking away, withdrawing something that we've had forever and ever, aspirin? Who would ever imagine that withdrawing aspirin for antiplatelet therapy trials could be an important question? And it is an important question because I said where we were at the time of designing TWILIGHT is the equipoise. Both ischemic and bleeding complications were important. We have potent agents with the best possible devices in high-risk patients coming in. Can we give that potent agent for a longer period of time if the aspirin is not there? What is aspirin doing in presence of a very potent antiplatelet regimen like ticagrelor? And I think that was a very, very key question about withdrawing aspirin. And it wasn't just me. There were many trials on P2Y12 monotherapy, meaning you start with the dual therapy, but instead of dropping the P2Y12 inhibitor, you now drop the aspirin. Who dares to drop the aspirin? Aspirin has been the mainstay of therapy for as long as we can remember. Certainly without aspirin, PCI, percutaneous coronary intervention, would never have survived. It was very, very important because stents are so thrombogenic early on, as I described. So there are many, many trials that have been there. I've put 4 of them. And of course, we're going to talk about TWILIGHT, which was in the New England Journal of Medicine 2 years ago in 2019. And let me tell you how we came about. This stacking is an underappreciated enemy. We have a drug that we're used to. We get new novel drugs like we had clopidogrel, then we went to prasugrel and ticagrelor, but we never thought about, well, do we drop aspirin? We just added it. And it's the same thing in all of these antithrombotic, anticoagulant therapies. We're often adding it instead of taking away something else. And I think what we hear from our statistical gurus of the world, Professor Pocock, in his editorial saying, do current trials meet societal needs? We need trials that investigate withdrawal of certain established therapies to see if such withdrawals are beneficial, harmful, or null. It's important. And so it was the big question, can we safely withdraw aspirin when there's a novel potent agent on board? So it comes back to the question, why is aspirin such a cornerstone of dual antiplatelet therapies? And I'll bring this to you because this is from 12 years ago in the Lancet, where they got 16 randomized clinical trials, 17,000 patients, and basically said aspirin is a lifesaver as a secondary prevention. If there's nothing else around, you could reduce major vascular events, coronary events, and strokes, and the price is not that big. The price is for every 10,000 patients that you treat with aspirin, you'll reduce ischemic fatal and non-fatal events by 100 to 200, while you will have 10 to 20 major extracranial bleeds and just 1 intracranial hemorrhage, that calculus works for that population. But where are we today? Today, the risk reduction is very different than where we were before. We have novel strategies, contemporary drugs that changes that risk profile of that patient such that that relative and absolute risk reduction of aspirin could be very, very different when you have all this other great drugs around like beta blockers, statins, PCSK9 inhibitors, SGLT2 inhibitors, heart failure therapies, all kinds of stuff that reduces that risk, that huge risk that the patients had in those other trials. We also have novel antithrombotics. Just as the stents improved, our antithrombotic regimens improved to reduce ischemic events. And so it begs the question, why do we need, if you have a novel antithrombotic regimen that works great in platelet aggregation and adhesion, why do you need a weaker platelet inhibitor like aspirin? And of course, the most important one is the bleeding risk of aspirin in combination with these novel antiplatelet therapies, but also its real direct gastrotoxicity that gives those GI bleeds that are tremendously important. And in fact, if you look at this trial that was already there way back when, we started to see that in placebo versus clopidogrel compared to aspirin plus clopidogrel in high-risk patients who were high risk for stroke and TIA, there was no difference if you added aspirin or withdrew aspirin. And in fact, intracranial bleeds were higher when you had aspirin on top of clopidogrel. Clopidogrel monotherapy was associated with less bleeding, no difference in those events. And it was the WUST investigators that for the first time withdrew aspirin. I remember when this trial came out, we were very, very nervous about this. How dare they take aspirin away in patients who just had a PCI with atrial fibrillation? But these patients had an oral anticoagulation on board. The combination of triple therapy increases the risk of bleeding. And by just withdrawing aspirin, they reduced bleeding and didn't show any issue in terms of ischemic events. In fact, it was better for those patients who had aspirin withdrawn. And when you look at the Plato trial, the trial that was designed by the sponsor to look at the superiority of ticagrelor versus clopidogrel in acute coronary syndromes, there was a reduction in cardiovascular death, MI, and stroke, but both arms had aspirin on board, and ticagrelor plus aspirin was beneficial in reducing cardiovascular death, MI, and stroke. But there was more bleeding, even though their major bleeding looks like their endpoint, their definition, they don't show a difference, but non-CABG related major bleeds were much higher. When you give a more potent agent, you will have more bleeding, and there was more bleeding there. But the most telling part of Plato was this slide of the aspirin dosing that had an incredible relationship with the efficacy of the combination of ticagrelor plus aspirin. These are all of the education we needed for us to think about TWILIGHT. So if you see from this slide, it was looking and showing that in fact, it was the low dose aspirin in combination of ticagrelor that was the most beneficial, and that all the benefit that was seen in Plato was with patients who had low dose aspirin. And in fact, those with high dose aspirin and ticagrelor had the highest and the worst events. And so it begs the question that if less is more here, less dosing, will none be even better? And so we, as the trial, as we were watching all of this and thinking about this, and myself with our incredible executive committee, had a hypothesis. We have high-risk patients. We were watching patients both in acute coronary syndromes as well as those patients with stable syndromes, but high risk for both ischemic and bleeding complications be going home on aspirin and ticagrelor for a year. And we said, what if we had a look and look at those patients who are at high risk, who complete a 3-month course of DAPT with aspirin and ticagrelor. Can ticagrelor monotherapy be superior for the next year after this in terms of reducing bleeding, but have not leading to ischemic harm? So think about this. It was a switch of the usual efficacy endpoints. The efficacy endpoint here became bleeding, and the safety endpoint here became the ischemic complications. Complete switch around of how we would usually design a clinical trial. So let me talk to you about how trials are put together. Usually we have an investigator initiated or sponsored trial versus an industry sponsored trial, and how are they different? Let me first talk about the industry sponsored trials, because those are the ones you're most used to. Plato was an industry sponsored trial. Plato was designed for the commercial interest of AstraZeneca to bring forth this new drug, ticagrelor, this potent reversible antiplatelet regimen, to show that it was better than clopidogrel plus aspirin in patients with acute coronary syndromes. So it's usually focused on development and approval of new devices and drugs. The investigators have, of course, the executive committee that are chosen by the sponsor have a role. It's usually a consultant role of the trial design, but they're not the only voices. The ultimate decision and the analysis and publication is taken by the sponsor, although the scientific integrity is quite high, because of course, the executive committee would not be engaged if it wasn't that way. The budget is really good because these are for industry, and the quality is extremely high because it meets the standards for drug approvals, which are tremendously difficult. Well, now switch gears to an investigator-initiated study and think about us as the investigators who wanted to test this hypothesis that I just gave you. Why would AstraZeneca want to engage in something like this? Well, it was to answer a clinically relevant question. That was the investigator. That was my question along with my executive committee. And it wasn't really about expansion of label, but more knowledge and very much centered around the patient, not product or device specific, but really asking a question that's relevant for us as clinicians. The investigators, we completely designed the trial without any input from industry, and we took care of the analysis, publication, results. We had full responsibility for the safety, quality, conduct. That was all by the investigator, the sponsor, which becomes the investigator. My institution stood behind me, and I was the sole holder of this very, very big task. But there are less resources and there's less funding. Does that mean that the quality is less? There's less monitoring? This is where TWILIGHT was. We got a funding, or a fraction of the funding of, let's say, what you would do in Plato, but we wanted to answer a question in a very, very good way. So we designed this study. We said, well, these are the high-risk patients. They're going home on aspirin and ticagrelor. Let's give them an open enrollment period of 3 months, because nobody would want to withdraw aspirin before 3 months. These are high-risk patients. Ticagrelor and aspirin is what is the mainstay for these patients. They want to give this to these patients. And now we want to randomize them after 3 months. If they did not have a major ischemic or bleeding event, and they were very adherent to their drug regimen, we would then randomize them to ticagrelor plus aspirin versus ticagrelor plus placebo in a double-blind, placebo-controlled fashion. I felt it was the most important thing that this should be double-blinded. Why? Because aspirin is widely available, because there's a lot of commercials out there about how it saves lives, and withdrawal was already such an important big question that people didn't really want to they were nervous to engage. And if they knew that they may not be getting aspirin, we felt like they would be, there would be some biases there. And so I felt like it was very, very important, and we all did as investigators. What do you do when you have to set up this trial like this? We needed to make sure that the blinding was not just in the design, but that the blinding continued to have firewalls in the entire organization of this clinical trial. So the medical leadership, that would be myself, our executive committee and steering committee, had firewalls all around us. We had absolutely no access to anything to do with the data, even though it lived right in the same institution. We had huge firewalls to data management, operational team, recruiting sites, all of those things, complete firewalls. We could not know anything about the data. On the other hand, there was also the only unblinded person was the independent biostatistician, who was working only with the DSMB, who had permission only to the electronic DSMB data, but very, very huge firewalls there to make sure that the blinding and everything is preserved. And very important was the Clinical Events Adjudication Committee, which was completely independent and outside of here, but again, blinded to the study drug so that they can do unbiased adjudication of events. We included patients at high risk for bleeding and ischemic disease, but what's really important about when you look at a clinical trial is who you excluded, because the results of this trial is not for the following patients, especially those who are in need for an oral anticoagulation, who had a prior stroke, or have a STEMI, or anyone who had a STEMI presentation. So this is really important. And the rest of these, of course, all here, if you can imagine, this is a potent agent. You're not going to want to give that in with a patient with a chronic oral anticoagulation, and that the whole idea of the prior stroke was a very, very big one that we didn't want to have come in here, especially if we were withdrawing aspirin. So very important to think about who's excluded. Always look at who's excluded when you look at a trial. So how did we come up with the endpoints? Well, we wanted to show, remember, the efficacy now became the bleeding endpoint. And we chose BARC 2, 3, and 5, and we can talk about what, there was BARC 2 is clinically relevant non-major bleed. So you might say, well, who cares about that? That's a soft endpoint. It's really BARC 3 and 5. But we needed to come up with something that we would be able to do. And of course, it was very important to have the power, for this key secondary endpoint, which was the safety, that needed to be non-inferior hypothesis in terms of the hard endpoints of death, MI, and CVA right after randomization up to a year after. So let's talk about those composite endpoints. What are the drawbacks of composite endpoints? And what are the great things about it? First of all, a composite endpoint is there. If I just went after mortality, I would need to have 25,000 patients in this trial. That would never be possible. So what do composite endpoints do? They improve your statistical power. They reduce sample size. They avoid the analytical challenges related to competing risks and multiple comparisons. But the very big but is the interpretation that can be very challenging and even misleading, especially if the components have different clinical impact. In other words, let's say, and I'm going to show you some examples that I got from my good friend, Dr. David Cohen. And if the endpoint is driven by something that's less severe, less important, and there are much bigger challenges, and you're going to see this in antiplatelet regimens, when you have different endpoint components that move in different directions. So in other words, putting bleeding and ischemic events in an antiplatelet regimen is a very big problem because they will go in different directions, right? If you have an ischemic benefit from a drug, but also causes increased bleeding, you are fighting in that composite endpoint in two different directions for a net effect that may or may not be meaningful. And it's very important is what those components are when they're in that net benefit of endpoints that fight with each other. So let's talk about the drawbacks. So here's the best case scenario when we added apixaban to stent versus placebo. The primary endpoint, which was death, MI, and unplanned revascularization, was in favor of the use of apixaban, which is another antiplatelet drug intravenously, very potent antiplatelet GP2B3A inhibitor. But everything was in the right direction for apixaban. So it made great sense. And look, bleeding is not here because if bleeding was here, it would go in a different direction. There was much more bleeding in patients who received apixaban. The usual case scenario is that, yes, you win, but you are driven by a maybe not as important a revascularization endpoint. So for example, this is a brachytherapy versus placebo, where you see cardiac death being similar, Q-wave MI being very similar, but it's the driver is reducing target vessel revascularization. So this composite endpoint should have maybe not had MI and death and should have just had TVR, and it would have won. But now you're basically claiming that the entire composite is positive when it's actually just being driven by the lesser important target vessel revascularization. But the worst case scenario is this one in the syntax trial, where cabbage was compared to taxus. And you can see here where what you're seeing in favor of cabbage in blue here is revascularization, MI, and death, but you're seeing stroke go in the wrong direction. So how is this working in terms of what's important to the patient? And are we really powered for that endpoint of stroke? Do we pay a lot of attention to it? We had to think about all of this as we were designing Twilight. So here's our assumptions, and I'll try to explain to you why we chose the bleeding endpoint as our primary endpoint, as the efficacy endpoint, because it was aspirin withdrawal, and why we put the ischemic events as the safety event endpoint and as the non-inferiority hypothesis, because it makes sense, right? We're not expecting that you would have less ischemic events. We just want to not see a signal of harm in the ischemic events when you're withdrawing aspirin, when everyone thinks you should be on aspirin indefinitely, and we're saying withdraw it. So that's a safety issue that we needed to understand, that we weren't causing ischemic harm while we were showing bleeding benefit. Very different kind of nomenclature. And why did we think like this? Because bleeding matters. And these days we are seeing that major bleeding with or without a transfusion is just as important as a myocardial infarction. And in fact, it attributes to more deaths. This is data that I worked on in an acute coronary syndrome trial, where we actually followed patients and saw that the early bleeding had a detrimental effect, the same as a myocardial infarction. And so when we made our assumptions, we talked about the superiority in terms of BARC 2, 3, and 5 bleeds, and we wanted to have the best possible power. We assumed a bleeding rate of 4.5% in the control arm at 1 year with ticagrelor plus aspirin, and about and we would have at least a 90% or 84% power if we had a sample size of 8,200 patients. But what would happen to our non-inferiority? What kind of power would we get for non-inferiority? And our assumption for non-inferiority was that we would have an 8% incidence with an average of 5% from DAPT, 11% from Plato. We got these two from the previous trials. So we averaged these two big trials and came up with the 8% in the control arm. And what we looked for, and when you do non-inferiority, you're looking for what kind of an absolute difference in risk would you accept? And for us, we said a 1.6% absolute difference. In other words, if it was 1.6% worse than 8%, we would accept that. That would become our margin for which we would allow the non-inferiority if it went below for that safety endpoint, not efficacy, but safety. And I think that was really, really crucially important. To me, this was the most important endpoint. We call it secondary, but to us, it was a key secondary endpoint. And when you go to stent thrombosis, I want to make sure you all understand how underpowered we are with stent thrombosis with the trial numbers that we had because of the assumptions that we had. So when we came to the power calculations, we came up with this 9,000 subjects to be enrolled. What did the other trials do in the P2Y12 monotherapy, the ones I showed you? Global Leaders was the largest one, 16,000 patients, all comers, and they had all of them are all comers. Global Leaders, the most powerful one with 16,000 patients, but a very, very interesting experimental arm of P2Y12 ticagrelor alone after a month, as opposed to us after 3 months. These two had clopidogrel alone after a month, and in TICO, it was ticagrelor alone after 3 months, similar to us. All of them were open label compared to us, the only double blind. We had enriched with high risk criteria. The rest did not. Our DAPT control arm was for 15 months. Remember, we started with a 3-month enrollment, 12 months post randomization, and very different than everyone else who had DAPT for 12 months in their control. And in fact, in here with Global Leaders, the control arm, everyone on the control arm went to aspirin monotherapy after 12 months. Our primary endpoint was bleeding. And here you see their blind for Global Leaders, it was a superiority testing for death and Q-wave MI for this experimental strategy. In here, it was a NACE, net adverse bleeding and ischemic events. In smart choice, smaller numbers, hard endpoints of death, MI, CVA, and non-inferiority. You imagine that I just showed you what you needed, 9,000 patients to have some level of non-inferiority for safety. Here with 3,000 patients, they felt they could achieve non-inferiority, probably based on assumptions. So think about these things when you evaluate trial versus trial. And in here, TICO, they had a net adverse clinical events, bleeding plus the major adverse events, very different than us, and they looked for superiority of this composite endpoint. This is not to say who's better, who's not. I'm just trying to tell you what we did to accomplish our task of answering a simple question of aspirin withdrawal when you have ticagrelor on board. Can you reduce bleeding without increasing ischemic harm? I wanted to answer a simple question, and so I designed a simple study with a simple endpoint, and I went for superiority because if we don't reduce bleeding, then what are we doing? So we enrolled 9,006 patients. We wanted to randomize 8,200. We only randomized 7,119, and you see that we did everything possible to get every single patient back for follow-up, vital status available, and we did a lot of work for the highest quality possible in terms of counting pills and in-person visits, etc. Look at our enrollment. This is very important. This is a global trial that went around the globe and enrolled patients. And in most studies, you don't get 36% US enrollment. We worked really hard and we used important strategies by starting in the US. Usually you start in Europe, then you come to US in company trials. We took the bite and went after the US participants and then opened sites outside of the US. And our colleagues outside of the US were amazing. and it's the same for our randomized population. We ended up with a very, very high-risk patient group, 65 years of age, 37% diabetics, 17% with chronic kidney disease, 20% with anemia, 65% with acute coronary syndromes. The vessels were multi-vessel disease, high-risk anatomy in 64% of the cases, thrombus present in 11%, calcific lesions, bifurcation, long stents. These are complex patients. And if we compare this to the rest, we see that we had a pretty complex patient population. We worked very hard on adherence. If you cannot keep the patients on the study drug, then your experiment will fail. It is not about what you end up with at the end. It's about adherence to the study medications you are testing in your clinical trial. That is the quality you need to strive for to get the right answer. And we did. And guess what? We had assumed a 4.5% bleeding rate in patients on aspirin and ticagrelor. We felt that the bleeders would come out in that first 3 months and not be randomized. But you know something? Even if you didn't bleed after 3 months, you ended up bleeding 7.1%. And we reduced that by a factor of an absolute risk reduction of 3%, numbers needed to treat 33. We proved that a double-blind placebo-controlled trial like this, aspirin was causing a lot of bleeding, and in fact, a lot of significant bleeding. We showed a 51% reduction in BARC 3 and 5 bleeds. These are significant bleeds. And all of the bleeding endpoints were positive for the ticagrelor monotherapy. But that's not the big question. Of course, you're going to reduce bleeding. I couldn't believe the magnitude, I tell you. But the biggest one is the key secondary safety endpoint. Our data safety monitoring board kept telling us, move ahead. We had 7 data safety monitoring board meetings during the course of the study, and they kept telling us, you may continue enrollment. And here's what they had been watching, that the endpoint, that safety endpoint, that's what they're there for, to keep our subjects safe, was there in terms of the hard endpoint of death, MI, and stroke. We met the non-inferiority margin. We didn't go to the 1.68. We were the upper limit bound stopped at 1.25. The curves were hugging each other throughout the time. There was no harm. And in fact, if you look at all of the endpoints, they looked pretty good, except for a very small 0.5 versus 0.3 in stroke, which is underpowered, still important. Here's TWILIGHT. Here's what we did compared to everyone else. If you remember, we showed superiority in global leaders. They, even though they had a good endpoint, they looked better. They lost the superiority. They didn't demonstrate superiority, became a negative study. In these other trials, they demonstrated non-inferiority and superiority. And when a network IPD, sorry, not network, an individual patient data gathering by myself and Marco Valgimigli and all these incredible investigators came through, we showed that yes, you will reduce major bleeding, BARC 3 and 5, when you go to a P2Y12 monotherapy without causing ischemic harm in terms of all-cause mortality, MI, and stroke. And in fact, we wanted to make sure that there was biological plausibility of this. We did a blinded platelet study looking at blood thrombogenicity in 40 subjects from the time they were randomized until 4 months after, with blood draws, when you can see how there was absolutely no difference in the blood thrombogenicity. There was nothing that aspirin was doing in terms of reducing platelet inhibition more than what ticagrelor was doing. And as a result, the ESC guidelines awarded ticagrelor monotherapy a 2A recommendation in a select patient population. And I think it brings us back to, how do you make the decision? Where are you and where are your patients? How do you design the trial? Are you at a high ischemic or a low bleeding risk? Can you put all of these patients and try to figure out something that comes true? And I think that for future trials, we may need to be focusing on where are these patients, and answering the questions that are relevant for those patients is incredibly important. I just want to thank everyone and go back, but I can't tell you how much this could never be possible without all of these investigators who helped us, and our collaborators that just will go on and on, that helped us bring Twilight to fruition. They're all listed, every coordinator, every investigator, and then most importantly, all of the patients. I think we came forth with a clear-cut answer in a double-blind placebo-controlled trial that when you have a potent agent on board, you need not have aspirin on top of it for that select patient group, because you will reduce bleeding when you go to ticagrelor or monotherapy without increasing ischemic harm. So thank you so much for your attention, and I now open this up for questions.
Thank you so much, Dr. Mehran. That was a real masterclass on how you came to design Twilight.
I'm sorry I took a little bit longer, but I had to give the full story.
No, it was fabulous, because I was reading Twilight this morning and all the questions that occurred to me, why you chose bleeding. I mean, that's such an atypical primary endpoint for ADAPT trial, why you chose that with the superiority design, and you explained it so beautifully. I just have one question, because when I read that and I read MasterDAPT and I see how they had 3 ranked primary endpoints, the first of which was, as you also discussed, net adverse clinical events. So what made you choose only bleeding as opposed to NACE, and how much more, I would assume, complex would that sort of trial design be where NACE were the primary ranked endpoint?
I mean, I think that if we had gone the easy route of NACE, and I don't want to say MasterDAPT is pretty unbelievable, 4,000 patients, high bleeding risk. I think, you know, for high bleeding risk patients, the net benefit is important, right? And I think that's an important variable. But we can always do that calculation afterwards without confusing the results by separating and seeing exactly if you have power for both of these important ischemic and bleeding endpoints, then you will answer the question in a much, much more concise manner, where you will be able to take on a conclusive statement about the results. I think that, you know, if we had chosen NACE, we would need much less patients. What degree of confidence would we have in the ischemic endpoints? It would be questionable. And I still say that even with the MACE endpoints that Dr. Valjamigli showed so beautifully in those high bleeding risk patients, they have a lot of ischemic endpoints, and they were lucky to show that they didn't pass the non-inferiority margin. Remember, non-inferiority is just about what you set that margin to be. I mean, how is it that the rest of our teams that they did these P2Y12 monotherapy studies were able to do it with so fewer patients? And why did global leaders need 16,000 patients? Because they were looking for the real hard endpoint of death and Q-wave MI, superiority. So, you know, the devil is always in the details on how you design these studies. But I felt like if I was going to do a study, I never wanted to question back and have everyone, maybe because I always have imposter syndrome, that thinking that, oh, you know, this is going to be really, really scrutinized. It's the first time we're withdrawing aspirin in a double-blind controlled, placebo-controlled trial. So I said, well, okay, we have to have double blinding. And of course, that raised $3-4 million to the budget, to the bottom line budget. Oh, it's aspirin, it's everybody, you know, why do you have to make that a placebo? I said, oh, because it's really important. It's because it's such a commonly used drug. And so the fact that no one knew what the patient got, that all was very, very helpful as well. And I think it was very important for us to keep those bleeding and ischemic endpoints separated, to be able to come back with a definitive answer for patients, for clinicians, for those who are writing guidelines, hopefully, et cetera.
Thank you very much. I'm inviting all colleagues who are watching to ask their questions in the chat room or on the YouTube comments. So, if I may, I would continue with one question. So, the secondary endpoint, which is the ischemic endpoint. So, in my understanding, it's actually not a secondary endpoint. It's you obviously had two trials in one. What was one superiority trial and one non-inferiority trial another, and both of them had one key primary outcome. So that's because
I wish that I could say that that was a co-primary endpoint. We, in our statistical analytical plan, we wanted to be careful based on our funding, because if it was a co-primary, then we would have needed at least 8,200 patients, and we had 7,119. And we had no idea of the 9,000 patients. We thought only 10% would fall off. Well, more than 10% fell off, as you saw. And then having that be a key secondary endpoint, but powered for non-inferiority with a margin, our power was not that perfect. The power needs to be a lot better than what we had with 7,119. So is this definitive as a primary that it's absolutely non-inferior? Well, you saw stroke was a little bit higher, right? And so, yes, the things looked great, but it could still be that we're not 100% there with the 7,119 patients. So I will never say that this is a co-primary. And because it's a safety endpoint, you could get away with a safety endpoint being a key secondary. The safety here is switched to an ischemic outcome of a composite. And it was basically saying that we would be efficacious by reducing bleeding, but we wouldn't cause ischemic harm as our safety. And because it's safety, then you could stick with a per protocol, not ITT. These are all the little things that you could do in a safety protocol. And of course, with the New England Journal of Medicine, as you know, secondary endpoints are no longer part of the abstract, but they allowed this because it was just such an important secondary endpoint. As you are saying, you think of it as a co-primary, but it really wasn't. It was a key secondary endpoint. And of course, we have to be transparent. I showed you all of the power calculations, exactly how we have it in our statistical analytical plan.
How many?
Sorry, Najawid, just
roughly how many more patients would we have had to include to be powered for the ischemic endpoint?
At least 8,200, and we had 7,119.
Mm-hmm.
Am I right to assume, Dr. Mehran, that if it only met superiority, would you then be able to test for non-inferiority? Is it that sort of hierarchical
No, we didn't
plan?
do a hierarchy. No, that was the other thing. So that's why it became a secondary, a key secondary. And it's key because it's such an important endpoint. And we did not have a hierarchical testing at all. No.
Absolutely.
Could you elaborate why you picked the 3 months?
Very good question. I mean, in retrospect, I think, 1 month is probably even better. And perhaps we would do extremely well as well. But you know what? It was the very first time. You can't know what it was like. I can just tell you, the Italian authorities gave us took us 8 months to get through Italy. It took us about 1.5 years to get through BfArM, that they would believe that withdrawal of aspirin could actually be done in a double-blind placebo-controlled fashion, that nobody would know, that we'd have to wait to the end of the study. There were all kinds of issues. What if the patients, and that was the other thing, is that we didn't want to be in a dangerous zone of stopping ticagrelor, putting patients on ticagrelor monotherapy, if then they were still maybe would have shortness of breath 2 weeks after you randomize them, and then they stop ticagrelor, and then there's nothing there in half of the patients. So we were very careful that, and we know that that first 3 months, the first 90 days is the most events happen in that first 90 days. So, and it's exactly around that time where you might want to make a change, whether you want to de-escalate, whether you want to shorten duration, especially in different patients. And you become more feeling a little bit more safer in changing your regimens. And so at the end, the 3 months is what we chose. In retrospect, I mean, I think, you know, if we're thinking about our next trial design, would be more inclusive, less exclusive, and probably include an earlier withdrawal. Because we saw wholeheartedly that it really was doing nothing in terms of thrombogenicity, blood thrombogenicity.
We learned that having trials on a global level, you know, which are enrolling patients on a global level, are super complex. They are very costly. They're very expensive. You stressed that it's a positive characteristic of this trial that it was globally conducted.
Yes.
Why do you think that?
I would say that the best thing that I ever did is I made, like, I don't know, 2,000 new friends, 2,000 new collaborators amongst our collaborators, our PIs, and the coordinators at every one of these sites. And if it weren't for them, we wouldn't be able to deliver this because you know, and I'll be very open with you, each site received only $1,000 for their entire duration of the trial for that patient enrollment per patient. The drugs were given for free, of course, the open enrollment, as well as the placebo, and the entire drug regimen was given to the patients free. But the sites only received $1,000. That's nothing in today. And so that is academic collaboration, passion, friendship, and the zeal for an answer of a very important question, and believing in a trial that is well designed, well conducted, and implemented. I could not have done this. You know, all of our colleagues in the different countries took on the leadership roles of going to their ethics committees in their local countries and taking on the costs of doing those. The number of the global collaborators were just unbelievable. We obviously had an EDC. We obviously had, you know, CROs in different places, but they were all run by academic research organizations that helped reduce the cost for us.
Thanks, Dr. Mehran. I just have a couple more comments. One of them is there was remarkable adherence as well as completion of follow-up in a global trial of this magnitude. How did you manage that? And one last question is about, because this is something that you ask everyone, and I feel the need to always ask everyone about the gender ratio of the enrollment of women in TWILIGHT also. Just a couple of comments on that.
So, you know, one of the enrichment criteria was female. I think you saw that on the criteria that we wanted to have a female, but still we only had 25% enrollment. And probably because we had exclusion criteria that excluded women. The need for an oral anticoagulant, that's usually AFib in women. Prior stroke, we see a lot of women with prior stroke. Cancers, all these other things, they were all part of so we only ended up with about 25% female, even though it was an enrichment criteria, and I wanted to enroll more women. Very difficult to also get them to come and collect their drugs and come in for their in-person visits. And we didn't have funding to make those things possible. So of course, in the future, there has to be a much better way to be sure that we have, enrollment of a diverse patient population. And this was back in 2014 when we started thinking about this trial. And of course, I've always, for the last 2 decades, I've been thinking about enrolling more women in clinical trials, and we did not succeed. And I'm obviously not very happy about that. Your other question was about how did I get adherence to the level that we did? Well, bringing the patients back in person with their pill bottles and counting their pills and then looking at their adherence and then educating them was another way of like making that connection with the patient, the importance of them taking their drugs, and then counting the pills. And if they were under, they were told, oh, come on. And then the next visit, we would follow them. It was tremendously helpful. So this kind of talks again about the televisiting that could have an important impact. Just some form of contact that's personable with that patient, even if it's on a video or a FaceTime call, could be extremely, extremely helpful in getting that patient to follow directions, to not only to adhere, but also to remain in the study. And I think that's retention is very, very important. If you look at, unfortunately, in global leaders, they didn't have, it was a pragmatic trial, and they just basically, and there was about a large numbers of patients that did not adhere to, and that could have been why they lost their superiority, because it looked like they were just, the p-value was 0.07. That's got to be very, very frustrating.
You're right. And your point is well taken about some of the exclusion criteria per se, excluding women de novo at the outset. Yeah. So what do you think we can do? And do you think, say, coronary artery disease trials like this by default have a role in excluding women in inclusion because of say the nature of the disease, or as you mentioned, some of the stipulated exclusion criteria? And what can we do then?
Yeah, so we need to kind of work, all of us collectively, in making, and I think we made a lot of recommendations in the Lancet Commission, which would be recommendations to myself as I'm designing clinical trials to be more inclusive, to have more education, to enroll women where they are frequented, and to make and remove barriers for follow-up and provide transportation, televisiting, however which way, because women have a lot of burden on their shoulders, and to ask them to do one more thing when they are doing 100 things, it's extremely difficult. So we have all of those things now for future trials to try to, and there is no reason why we shouldn't be able to do this.
Thank you very much. That was awesome. Thank you.
Thank you very much. I think Aisha and me, we could continue for hours.
Me too. Thank you.
And especially, I had in mind to ask you what you would derive from DOAC trials, not only compare this to the risk of other antiplatelet therapies, but maybe we can pick, if you have a sentence on this, I would be happy about it.
Yes, no, I think very important. I showed the WUS trial because it was the first that withdrew aspirin. And then with it came a whole slew of trials that looked at less is more. In oral anticoagulant patients, less is more, but not one size fits all. We should really talk about that. And at the moment, we are looking for that perfect drug, maybe that works on contact thrombosis, where we could have the least ischemic events with no bleeding harm. And those are some of the trials that we're now designing in anticoagulation realm with factor 11. And I think this is really, really very, very important. And in the DOAC trials, as you know, for the most part, the withdrawal of aspirin with the novel oral anticoagulant is superior in terms of bleeding without ischemic harm, but none of them are powered for the ischemic harm, not even in the way that we powered our study, unfortunately. And most of them, the withdrawals are open label. Except for Augustus that had a double-blind placebo-controlled aspirin withdrawal.
Thank you very much. I hope we can invite you again in a
I
not so
would
far
love
future.
it. Thank you so much. Thank you both for having me. I hope I did not disappoint, and I hope that people took away some of our rough experiences with Twilight. It really was, it felt like there was twilight, and then the sun came. So thank God.
Oh, that was fabulous. We learned so much, and you answered so many of our questions. Thank you so much.
Thank you so much. Thank you.
Thank you very much.
Bye-bye.
Hope to see you soon. Bye-bye.
Bye-bye.
Bye.